Hello everybody, my name is Sabine Seidel. I'm postdoc at the Crop Science Group of the University of Bonn, and today I will talk about roots. The Green Revolution of the last century boosted crop yields in developing countries by introducing draft genotypes tolerant of low soil fertility. Another major achievement was to maximize the harvest index by converting biomass into grains. The first Green Revolution was based on crops responsive to high soil fertility, but the new Green Revolution will be based on crops tolerant of low soil fertility. However, modern crops may have lost beneficial root traits for low-input agricultural systems. This figure shows the root length of 9 wheat varieties released in Australia between 1958 and 2007. You can see that the root length over soil depth decreased with the release years. The oldest variety showed the highest root length. That means 5 decades of selection for yield reduced the root length density. The roots will play a major role in the new Green Revolution. Optimal cultivars tolerant of low soil fertility and drought are often described as deep rooting with an intensive root system. But wouldn't an increase of the root system result in yield losses? But let's take a step back again. Plant traits are the morphological, anatomical, physiological, biochemical, and phenological characteristic of plants, and they determine how plants respond to environmental factors. Certainly you know some shoot traits such as the leaf area index, leaf nitrogen content, biomass, leaf angle, grain number, and others. I think you may also heard of some root traits, including the root length density, which is the root length in centimeters for a defined soil volume in cubic centimeters, or the root biomass, the root diameter, root angle, and some others. For many classical root traits, the correlations are well known. The figure on the top right shows you, for example, the correlation of the shoot dry mass with the leaf area index of winter wheat. And on the bottom right, you can see the correlation between grain yield and plant height for the same dataset. Also, we often know how to interpret shoot traits. Look, for instance, at the two pictures on the top left. They show 2 sugar beet fields which were treated with different fertilizers. The sugar beet plants on the top left perform much better, and most probably also the yield will be much higher as compared to the right picture. The figure on the bottom left shows you winter wheat grown under N-deficient conditions. So it's even visually obvious that the plants on the bottom and on the top right don't perform very well. However, with root traits it's more difficult. If you observe a certain root length density distribution over soil depth, it's hard to say something about the crop performance or to correlate it to yield. Have a look at the 2 root length density distributions in the figure on the right side. They could differ because we compared 2 different genotypes, or the root length density in case of the blue one could be higher in the topsoil because there is more phosphorus in the topsoil. It could decline at 40 cm soil depth because there is a compacted layer, which is not the case for the red one. And the root length density in the subsoil could be higher because the soil is dry in that treatment, or just because the total biomass is higher. In the first part of this lecture, I will explain you which factors influence root growth with a focus on root morphology. First of all, the genotype of course influences root traits. Plant breeders are generally hesitant to consider root traits for selection because these traits carry a low heritability, they vary a lot across environments, and the field measurements are very labor intensive. Root traits under genetic control include the root angle, which I will address a bit later, the root preference for either rooting shallow or deep, and lateral branching. And the variation in root traits can be really large. Think of a field trial, for instance in Campus Klein-Altendorf, where the yield varies maybe for between 8, 9 and 10 tons per hectare. In this study, scientists compared the root angle of barley in 20 feet trials. The root angle is shown here on the left side, exemplarily for one shallow and for one steep root angle type. And in this study, the root angle varied between 12 and 89 degrees. Roots are also strongly influenced by physical constraints. In dry or compacted or both soils, the resistance to root penetration can be quite high. In case of high resistance and a susceptible genotype, the root system changes. The root length and number is reduced, resulting in a smaller root system. The diameter increases, the root angle gets less steep, and root growth is deflected. Also, drought and dry stress influences root characteristics. Genotypes vary in the timing of the active root growth. The root systems are more vertical, more branched, the root length in general is higher, and the plants and develop deep root systems. Some studies report about thinner, others about thicker roots. Some crops form a so-called root cortical arrangement, which reduces root respiration by converting living cortical tissue to air volume. This seems to increase the drought tolerance by reducing. But is a deep root system always beneficial? I would like to show you a nice study from colleagues that compared durum wheat genotypes in two different environments, and they divided them into deep, and here shown in the red bars, and shallow rooting genotypes. The top panel shows you the grain yield under rain-fed conditions for two sites, and as you can see, the deep rooting genotypes perform better. The authors conclude that the durum wheat grain advantage was about 38% under low moisture conditions, and this was mainly achieved by the deepest rooting genotypes. But now let's have a look at the irrigated treatments, shown at the bottom panel. In case of irrigation, the deep rooting genotypes performed worse as compared to the shallow rooting genotypes. The yield penalty was 20 to 30% over the whole three environments, compared with the shallow root systems. After addressing the effect of drought on root characteristics, let's now have a look at the effect of nutrient deficiency on root characteristics. Hereby, I will only address nitrogen and phosphorus P and potassium K. This figure was generated by Gina. She writes a review about the effects of nutrient deficiency on root growth, root morphology, and root-to-shoot ratio in common arable crops under feed conditions. And she analyzed the results of about 50 papers. The graph shows the effect of nutrient deficiencies on root traits, and it shows it as a relative change. On the left side, it shows the effect of root length, then on root biomass, then on root length per shoot biomass, root-to-shoot ratio, and the right side on specific root length for the three nutrients N, P, and K. The results suggest that the root length and root biomass decrease in case of nutrient deficiency. This is true for the absolute values, and the main reason is that also the shoot biomass is reduced. However, the root length per shoot biomass and the root-to-shoot ratio are enhanced in case of nitrogen and phosphorus deficiency. In short, the absolute root length and root biomass decrease in case of N and P deficiency. However, the relative ones, considering also the shoot biomass, they increase. What else influences the root traits are the sowing density or the competition. In this study, different sowing densities of feed-grown spring barley in the plant row on root length density were tested at Campus Altendorf. As you can see in the figure, the root length density increases with increasing sowing density. The higher sowing density also led to more shallow roots, through increases in topsoil fine root production. Besides that, the tiller and nodal root number per plant decreased with an increasing sowing density. On this slide, I would like to show you some work Sophia Hardy is doing. Last year, we took some root samples in the mixture trial at Campus Altendorf, where springweed and faba bean are grown in an in-row mixture. That means the plants are growing next to each other in the row. We took several soil samples on 2 dates, and then we processed the soil samples, and then we did we used the FTR spectrometer to discriminate between the species. And I would like to show you one result in the graph at the bottom. The box plot shows the root length density in the depths of 70 to 100 centimeters of several different treatments. On the very left side, the bar shows the root length density of faba bean only, and on the very right side, it shows the root length density of one of the springweed cultivars. There was also a treatment where we just added up the same treatments. That means we had the same amount of faba bean plus the same amount of springweed. This is the treatment that is presented in the second left bar. As you can see, the root length density in the mixture is between the one of only faba bean and only springweed. It doesn't sum up. In contrast, for the shoot biomass, which is not shown here, this is the case. So the shoot biomass, in case of the mixture, the high dense mixture, is higher as compared to the faba bean and the springweed only treatments. One explanation for this is complementary root growth. The roots may use the resources, but not at the same time and at the same place. The roots, of course, depend on the assimilates that are produced in the shoot part of the plant. And the roots are responsible for the water and most of the nutrient uptake. So the easiest way for a plant to get more nutrients and water would be to have a large root system, and thus a low shoot-to-root ratio. However, bigger root systems take more carbon away from the shoots, and this limits the plant's capacity to fix and store carbon in the shoot and also in the harvested yield. This is obviously a trade-off. But can we correlate root traits to yield? Root growth is a dynamic process that involves complex interactions among the atmosphere, the plant, and the soil. Therefore, it often becomes difficult to trace a meaningful association of roots and yield. You can see here correlation matrix plots of winter wheat traits from the long-term field experiment DECOPSO from 2020 for 2 dates. On the left side for mid of March at tillering, and on the right side at mid of May around flowering. So we observed the root length on the top, root dry matter, grain yield, shoot LAI, and at the bottom the plant height. And as you can see, already at the early growth stage of tillering, there is a strong correlation between leaf area index LAI and the shoot dry mass of the plants. Also, the grain yield and the shoot biomass and the grain yield and the leaf area index correlate. Unfortunately, the correlation between root length and yield, or root dry matter and yield, is very low. Now let's look at the correlation matrix plot on the right side at flowering. Again, the correlation between the shoot dry matter and LAI is very high. Also, the correlation of grain yield and LAI and plant height and grain yield are rather high, so they are red, deep red. And now at flowering, where the root growth is supposed to end, there is some correlation between the grain yield and root dry matter and root length, though it is not very strong. I have to admit, I'm not a breeder, but I couldn't leave that topic out. What we learn from literature is that the root architecture is likely to be multigenic and thus much more difficult to select for as compared to other traits. Breeding for root characteristics has seldom been implemented. As I have already mentioned, the traits carry low heritability and they are variable in expression across environments, and also the feed measurements are very labor intensive. I've shown you in one of the studies that deep-rooted durum wheat genotypes yielded higher in dry environments, but they yielded lower in the irrigated treatments. There are also further studies that indicate the relation of a certain root trait, such as root length or root thickness, with grain yield. In the first part, I explained to you what influences root traits: the genotype, water and nutrient availability, soil compaction, and the sowing density or complementarity. In the next part, I will raise a bit different question. How should root systems look like for a defined environment? Let's start with compacted soil layers. How should root systems look like when they grow in compacted soil layers? What is favorable to gain high yield? In general, genotypes with a narrow but strong root system, an increased root diameter, and a high tortuosity perform better in compacted soil layers. And what's favorable in case of drought stress? Growth vigor in the early vegetative stage, but also longer and thicker roots with a higher root dry weight prior to stress are favorable. Some studies report of a better behavior of the genotypes with a reduced lateral root branching or with narrow xylem vessels to conserve water. Several studies also report that deeper rooting types, especially in terminal drought, are favorable. So in general, the steep, cheap and deep root architectural ideotype is favorable in case of drought stress. Why? Because it allows a greater acquisition of water during drought as the water table drops. And how should root systems look like in case they are grown in low nitrogen conditions? Again, a rapid root growth at the beginning of the plant development is favorable. Some studies report that it's favorable if plants have a shallow root angle at the beginning of the growth stage, but a steeper one later. Also, several studies report that a large root biomass, but also an increased root length density and root mass in deeper soil layers is of advantage. Besides that, the increased formation of a root cortical parenchyma or also interactions with microorganisms that enhance the availability of nitrogen in the soil are favorable. And what about low phosphorus conditions? Again, a good growth at the beginning of the stage of the growing season is favorable. In contrast to low nitrogen and drought stress conditions, in the case of low phosphorus conditions, a shallow rooting system is more favorable. But this has mainly to do with the fact that phosphorus that is applied as a mineral fertilizer is usually put on the topsoil, and then the highest concentration of phosphorus is in the topsoil. Well, we don't have to go now in detail through the whole list, but I want you to understand that some of the characteristics are similar to the ones we have heard before in case of N and drought stress, and others are not. I also would like to point to a study that is shown here on the right side from Miguel et al. And he invested several common bean varieties with different root angles and root hairs. And the main finding of that study was that the variety with a shallow root angle and long root hairs led to almost 300% increase in plant biomass on the low phosphorus soil. Let's sum up what we have learned until now. Different root ideotypes have been developed, for instance for low N, low P or low K or low water conditions. An ideal plant type for a certain climate or environment is referred to as an ideotype. Crop ideotype is a plant model that, when developed as a cultivar, is predicted to generate a higher yield. This scheme here shows three different ideotypes. On the left side for low P conditions, in the middle for low K conditions, and on the right side for low N and for dry soils. But what do they have in common? The characteristics they have in common are the early stage vigor, or the rapid root growth at the beginning of the development, a large root biomass or root-to-root ratio, high root length density, also the ability to root deeply, and a large root surface area. However, many of these ideas have not been realized through breeding programs, as I have already mentioned. In the first part of the lecture, I explained you how roots change mainly their behavior, morphology, and physiology in response to changing environmental conditions, such as compacted soils or low N conditions. In the second part, I explained which root characteristics are in general favorable for several environmental conditions, such as compacted soils or low N conditions. But how should root systems look like in a changing or in an a priori unknown environment? Of course, low P and low soil water conditions are possible. Or imagine you want to grow spring wheat in a field nearby Bonn. Will it be dry, such as in the growth period in 2018 and 19 or this year? Or wet, such as in 2021? I would like to mention two options. The first one is root plasticity. The phenotype is the product of genetics, environment, and the interaction between genetics and the environment. Phenotypic plasticity is the ability of plants to alter their behavior, morphology, and physiology in response to changes in environmental conditions. As you have seen, several biotic and abiotic constraints limit root growth, and phenotypic plasticity is an important phenomenon to enable plants to adapt to their environment. Root plasticity has been proposed as a target for the development of more productive crops in variable environments. Root plasticity may be beneficial in natural or low input systems. However, in high input agroecosystems, the value of phenotypic plasticity is unclear. The graph shows examples for more and less plastic genotypes. It shows the relationship between total root length and soil moisture content. The authors compare 4 rice genotypes. We have learned before that an enhanced root length is favorable in case of dry conditions. And an enhanced root system and deep roots are a waste of carbon in wet conditions. For 3 of the genotypes, the total root length increases with increasing soil moisture content. However, there is one genotype marked in red which behaves differently. It shows a high root length in case of low soil moisture content and a rather low root length in case of high soil moisture contents. The authors of this study concluded that the plastic root development was the key trait that contributed to yield through increased total root length and thus water uptake. Another way to deal with variable environmental conditions are variety or genotype mixtures. In this case, not one variety is sown in one field, but 2 or several ones. Variety mixtures have several benefits. Resource use complementarity is an expected key underlying mechanism, provided that the varieties in the mixture differ in resource-related root traits. One could, for instance, combine more shallow rooting with more deep rooting varieties. Another advantage of variety mixtures are less pathogens. In general, mixtures of varieties typically are more productive than monocultures of the same variety. However, this overyielding is often limited to a modest range of only 1 to 3%. Some variety mixtures, for instance of maize and of spring wheat, were approved for cultivation by the Bundessortenamt. However, commercial efforts to develop, release and promote variety mixtures remain peripheral. Additionally, models can help us to identify root ideotypes. In general, data from field experiments or other data, for instance from greenhouse experiments or from literature, are used to calibrate and validate process-based crop models. These models simulate various processes including phenological development, light interception, root growth, soil, water and nutrient dynamics. The models can then be applied, for instance, for improved process understanding, to quantify ecosystem services, or, of course, to predict yield under different management scenarios or, for instance, future climates. The screening of genotypes and modeling approaches can also be linked to identify root ideotypes for different environments. That's the so-called in silico breeding. Not only the root growth is simulated on field and larger scales, but also yield and ecosystem services, such as the soil water availability or carbon storage. Models can assist us in finding answers to questions such as how should root systems look like for various environments, for instance under future climates or low nutrient conditions, and for various environmental scenarios. Some colleagues and me, we plan to link in-field screening of genotypes in own experiments or using available data of others with modeling approaches to identify root ideotypes for different environments. We want to simulate yield, but also ecosystem services on field and larger scales. The heart will be a field scale model coupled with a 3D architectural root model. We did some preliminary work on this topic, which was published this year. We coupled a 1D field-scale crop model running in the simplest modeling framework with the 3D architectural root model CRootBox on a daily time step. Additionally, we implemented a stress function to simulate root elongation as a function of soil bulk density and metric potential. The model was tested with feed data collected during 2 growing seasons of spring barley and winter wheat grown at Campus Altendorf. The 2 panels on the top show the simulated daily root length over soil depth of spring barley in 2018 for the deep loosen treatment on the left side and for the control on the right side. And as you can see, the root length density is enhanced on the left side where the soil was loosened. The 2 bottom panels show the simulated crop water uptake over soil depth and over time. And as you can see, the water uptake was lower in the control because of the soil compaction. And this soil compaction, or rather enhanced soil bulk density, decreased the root growth. With this example, I want to show you that models can help us to estimate values such as the daily root length density and crop water uptake over time, which are very difficult to measure. In the study, we also varied some root traits to find out which ones foster high yield and yield stability. We concluded that our model is a feasible tool to assess the value of physiological traits in targeted environments and for specific management, in this case for subsoil management. Let me summarize the key messages of this lecture. You learned that you cannot interfere the performance of the plant or the final yield just by looking at the root length density. In general, drought stress promotes root growth, especially in the deep soil layers. Nutrient deficiency often limits absolute, but not the relative root growth. Scientists have already described root ideotypes for defined environments. However, it's not easy to describe root ideotypes for variable environments. Models support the finding of ideotypes. One of the main bottlenecks in root science is that the work on field scale is very laborious and there is a lack of high throughput and large-scale phenotyping methods. With this, I would like to thank you for listening, and I'm happy to take your questions.